We have discussed about the biochemical model, and it was proposed in the 1920s. And this was a model which didn't get some serious attention of the other scientists in the 1920s, 30s, 40s. But in the 1950s, there was a graduate student, the Stanley Miller. He was a student at University of Chicago, and he made a model of Precambrian atmosphere and ocean in a laboratory glass vessel. So this student, who is at the time, the Stanley Miller, he made a model of Precambrian environment. What was the environment? We'll be discussing that. So he exposed a mixture of water, nitrogen, carbon monoxide, and nitrogen to electric sparks in a way that there was heavy lightning on the surface of the earth. And he found a brownish sludge in the bottle after a few days. So he produced this mixture, gave some sparks, left it after some days he saw a brownish sludge in the bottle. And now the story gets interesting. What was in the brownish sludge? In the brownish sludge, there were sugars, amino acids, and nucleotides. And we see these are the building blocks of the life. So was he able to create the molecules which were the building blocks of the life? And provided that he had some environment created of the Precambrian era, which was having such environment already on the Earth. So if Earth had those conditions, were these molecules were able to create? Earth was able to create these molecules on its surface? The researchers considered that the mixture of gases that Miller used was strongly chemically reducing. It was a possibility that although these gases were present on the surface of Earth, but these gases were not composition-wise as extreme as he used in his experiment. For example, he used the hydrogen, which was maybe present in the form of water, right? So, which is an oxidized form of the hydrogen. So there were some further experiments in the 50s and 1960s led to the production of polypeptides, polysaccharides, and the larger organic molecules. If you remember that previous diagram, the previous topic, in which these are the three steps, up till the polysaccharides, you have the third step. So biochemists were able to create those steps, and these were the further experiments which were able to somehow establish that, okay, these are some conditions which can result into the creation of some organic molecules. And then there was a discovery by the Sidney Fox. Sidney Fox was a student, a professor at FSU. He succeeded in creating the cell-like structures in which a soup of organic molecule become enclosed in a membrane. So he made some organic structure, which are, having some lipid membrane around it. And they were able to reproduce. They were, taking some nutrition. They were dividing themselves, but they were not able to survive very long. So this was a very, very, primitive protocell that was created by the Sidney Fox.